We can move on to our next speaker in the webinar, please. That's Jason Banda, and he's our early career spotlight from the University of Nottingham in the UK. Jason, if you're able to share your screen, please. It's come through now.
Okay, well, thank you,
Thank you.
Charlotte,
No
for the
worries.
introduction. I'm Jason Banda. I'm a postdoc at the University of Nottingham. And I just, first of all, would like to thank the AAB for allowing me to give this talk here to all of you. And today I really want to talk about the role of post-translational modifications, like simulation, and their role in plant adaptive responses, and especially today on drought. So Eva already looked nicely at nutrient acquisition, and I'm going to look at the other part of the equation, actually the water in the soil. So I don't have to remind the audience here how we're facing climate change at the moment, how we get more frequent drought and more intense droughts as well. And as you can see from the map here in Europe, it has a severe effect on the amount of moisture, the amount of soil moisture in the soil, and especially in Europe in this map. But everywhere we face this problem everywhere around the world. So we need to make plants. Adapt plants to maximize the influx of water from the soil, and of course to do that by the root system. So as you can see here from this beautifully CT soil core, on a macro level, soil exists out of a couple of components. That is the soil particles, the water around these soil particles, and air gaps in between the spaces where there is no water. And of course, these vary based on the type of soil you have, the levels of water within the soil. You can see it if there's a lot of water, then you won't have any air gaps. And the opposite, if there's a lot of dry soil, we'll see more often, there will be more air gaps and less water attached to these soil particles. So when a root is growing and trying to maximize the influx of water, it's going to come across all these different environments. And roots have actually evolved some very cool adaptive responses that allow them to look for water within the soil. So one of the most studied one, an early studied one as well, is root hydrotropism, where the main root or the primary root grows towards available water access. A more recent studied one within the University of Nottingham as well is zero branching, where they're looking at inhibition of branching in this so-called air gaps. So where there's no water available for the root can actually block the formation of laterals, and this will save a lot of carbon and other fuel sources that would otherwise go to waste if you branch towards somewhere where you cannot pick up any water or nutrients. And then what I want to really mainly focus on today is lateral root hydropatterning, where the branching happens only on the side of the root that is in contact with the moisture. And these are all driven actually by, it seems like it's by different molecular mechanisms. So that's quite interesting how they vary in that kind of the system approach. So what is actually lateral root hydropatterning? Well, it was first really described in a PNAS paper in 2014 from Jose Dini's group. And you can see from the images that were taken in the CT scanning in the Houndsditch facility at the University of Nottingham by Craig Sturrock. You can see that the influence this has in a real life soil. So on the left here, we can see maize growing into a soil column. And you can see if you look at the laterals, they branch out from everywhere along the root axis, so the root circumference. So everywhere where the soil has contact with water, there the roots branch. On the opposite here, on the right side of the screen, we can see if we build a macropore in the middle of the soil column, where this is like an interface between air and soil, we can see they only branch towards the moisture within the soil. This is lateral root hydropatterning. And actually, this story has come a long way. So again, this work from Jose Dini's group, where I was also involved in, showed beautifully what you can do if you have a diversified panel of maize lines. So I looked at a panel of 250 different maize varieties, and they checked how strong is actually the hydropatterning response in all these varieties. So if we only focus here on the air side lateral roots, so the roots actually and technically are not as useful, we can see that most of the maize varieties have about 95% or even more of their laterals pointing towards the water. So they're very strong selection pressure on finding water, which is of course logical. But there's actually a lot more variability than we originally expected. So some of those species, the varieties, actually have between 30 or 40% of their laterals towards air. So if you look, this is also beautifully visible on the CT scan images again. To look a bit different here with the soil being deleted out of it. But the concept is the same. You have in the middle a macropore, and on the sides you have the soil. And you can see on the left side of this, the strong hydropatterner. You can see all the laterals go towards the soil and away from the macropore. And in the weak hydropatterning, which you can see a lot of these laterals actually going into the macropore. So there's a proper selection pressure ongoing on here. And if we look at more in depth, like what varieties are actually like hydropatterning in what way, we can see a lot of these varieties, the tropical, subtropical varieties, were found as being very strong hydropatterning ones. So they have a selective pressure that really created lines that need to find water and maximize the branching towards this. While on the other side, the weak hydropatterning lines are actually the temperate varieties that potentially have relaxed their selection pressure on this adaptive response. So in the lab, I don't personally work on maize. I look more at the molecular mechanism that regulates hydropatterning. And I work on Arabidopsis, the model species, and look at the plate-based assays to find out where these laterals emerge from. So if they come out towards the air side or the agar or water side. And we can see that Arabidopsis is not as strong a hydropatterner as maize is. So we have about 80% going towards the water instead of 95. And there is actually a lot of variety between different species. So what we found out in 2018, that transcription factor RF7 is really important for this process. And maybe you already know RF7 is an auxin response factor and is very important for induction of lateral roots, both in initiation and development. So on the left side, you can see Columbia 0, the wild type Arabidopsis, and on the right side, you can see RF7, and you can see the reduction in lateral roots. So we hypothesized in this paper that on the wet side, you have RF7 acting as normal, inducing signaling events that leading to lateral root development. However, on the dry side, we found out that simulation is very important of RF7. So what is simulation actually? It's a post-translational modification, basically a protein modification that allows the switching on and off of proteins. So in this case, we can see that RF7 was bound by SUMO, or something we call simulation. And this allowed the repressor protein IA3 to come together with RF7 and block its functioning. So we have basically an on-off switch that is only based on either the attachment of SUMO or the detachment of SUMO. So it's a very nice engineering approach for engineering protein activity. So we have this on-off switch by simulation, and we could actually find that if we look at the protein domain structure, we could actually find 4 SUMO sites spread around the protein, all in different domains. And if you look at the conservation of these, for example, in barley, maize, rice, and wheat, we can see that they're also quite highly conserved, some of them more than others. So they could potentially be hotspots really for protein modification that could drive activity of proteins or maybe deactivate certain proteins if necessary. So I was actually interested in looking, because I'm from a more like a fundamental background, I was actually looking into how far did we actually come with understanding SUMO in crop species. So I actually found quite little on some of the crop varieties like barley and wheat. So there's definitely more to be explored here. But for example, if you look at rice, a lot of work has been done on that by trying to translate what we know from Arabidopsis into rice. So we know its importance in salt and drought stress. We know it's important as well in the nutrient acquisition, but there's actually a lot more that we need to discover first. We have a lot more fundamental knowledge that we can translate at the moment. So there's still a lot of scope here to get more translatable research. So I want to take you a little bit to the SUMO cycle because that's very important for the last part of the talk. So SUMO is actually translated as a full-length protein, and that needs to be matured before it can be activated. So these kind of like ULP proteases cut off the end terminal of the protein, allowing it to bind to an E1 activating enzyme. This then can become conjugated to a target protein to the functioning of E2 and E3 ligases. This is the point where you've seen the example of RF7. So the SUMO is attached to a target protein, and it can change the subcellular localization of a target protein. It can change the activity of a protein, or it can change even the degradation state, or even the interaction partners. So a lot can happen there. And of course, it's a SUMO cycle, so it's cyclical. That means also SUMO can actually be removed by these ULP and DESI proteases as well. And that frees up the SUMO pool and deactivates the changes that have been made by the SUMO. So in Arabidopsis, we have 32 genes or proteins that are important for this cycle of simulation. And we are very interested in our group to find out why actually do we need asumos? Why do we need so many proteases? They probably have functionally diversified. Otherwise, why would you have so many of them? So that is where the next part of the story comes into play, the SUMO cell atlas. So what we did is we marked all of these proteins with fluorescent tags to find out where they are expressed and localized within the root tissue. So you can see we use a specific construct in this case. And here you can see it as part of the E1 enzyme. So this construct allows us to look at both the expression of a gene and the protein localization. This we call the PMDS1 line. You can find all of this and more information in the recent publication from last year. And we basically, you can see that in the case of this E1 enzyme, you can see all of the nuclei, where the nuclei light up, basically that's where the protein is being transcribed. You can see the protein localization in yellow, and you can see it's also a bit hazier than the turquoise channel. That's just because the protein is also localized outside of the nucleus in the cytosol. And then we have we have the cellular solution by adding a red membrane marker as well into this mix. So this allows you to look at tissue-specific changes. So this we then used to look at particular different stresses. We can't, I'm afraid, we had 32 lines. We couldn't do all the stresses. So we just focused on mannitol as a sort of osmotic stress. Then we took ionic stress using salt. And we also had a pathogen stress by using flagellin. And you can see the variation that actually creates. So I'm going to pick up one example specifically about this. But first of all, I want to also highlight that all this data, so the base SUMO code data, so basically on the normal conditions, the expression and protein localization, and also the stress atlas, they're all on this Bar Uteronto website. You might have used ePlant before. This is basically a side page of that. So have a look and see if you find anything interesting genes for yourself. And I just want to show you like why is this important? Because for example, here's an example of a gene that is actually induced under osmotic stress conditions. So 3 hours after transferring it to mannitol, we can see a clear induction in the DESI2B. So this is a protease, so it takes off the SUMO from a target protein. And here on the top corner, you can see transcription in blue, again, and the protein abundance in yellow. And you can see a clear increase in the level of the signal after 3 hours of stress. So that means that DESI2B is actually being induced after osmotic stress. And it was not the case in all these stresses, or not in, I think in salts it was also, but not in the pathogen. So it's a very specific way of activating certain SUMO pathway components to drive developmental responses. So we're still looking into, for example, which components came up and which were induced and which were reduced, and what actually their functioning is. But this could be like a first step into creating an idea of which components are important in osmotic stress and why, and how can we translate this into the ortholog in crops. So with that, I would like to start summarizing the talk. So I hope I showed you the importance of root adaptive responses, and actually some of these adaptive responses like hydropatterning have been driving breeding selection. I would also like to hope that I convince you that SUMO is actually also a very neat model of a rapidly changing protein activity. And it is really crucial for what we've seen here in the case of lateral root hydropatterning for plant adaptation. And then also, I hope you will have a look at the SUMO cell atlas, which is a nice little resource that is now online. And it gives us a first idea of why certain SUMO machinery components are important in particular stresses. And with that, I would like to thank all the people who work on this. So this has been part of a consortium of people within the SUMO code. That is a BBC project. My mentors were at the University of Nottingham, Malcolm Bennett and Anthony Bishop, but it was led as well by Ari and Miguel in Durham. Some of the beautiful imaging you've seen the lines were generated by Deepan Shraboni and Sumesh in Durham as well. In University of Liverpool, we had a lot of help with the bioinformatics by Andy Jones and Ishan. And at the University of Cambridge, we have Catherine Lilly and Kishore doing a lot of the mass spec. And of course, I want to thank Jose Dini and Johannes for that amazing work on the wheat varieties. And with that, I would like to thank you all for listening and take any questions.